Hello everyone, my name is Rupinder Syal and welcome to Spartan Tutorials. Today we are going to talk about a serious threat that has been spreading rapidly around the globe and especially in India, and that is the COVID-19 double mutant variant. This has been recently isolated from sequencing of a limited number of samples, mostly from Maharashtra, by a sequencing consortium that has been established pan-India. And we have a limited data, but let's go into the biology of this double mutant variant and what to do about it. Now, the data that has been recently received by the Indian Sequencing Consortium on SARS genomics has revealed that the double mutant strain is the most common strain in India now. Although the data is pretty limited because the sequencing is not keeping up the pace with the number of cases that have been reported and that are climbing every day. We are now getting 1 lakh to 2 lakh cases per day. So the sequencing of those samples is not keeping up in pace with those cases, but still it appears that double mutant is spreading very fast, and it is spreading even to other countries. So Guardian recently reported that actually it has been reported in UK and locations as far as California, and this is due to obvious reasons because recently travel restrictions have been relaxed and people have been traveling back and forth between different countries, so it has spread to many different countries. In fact, the outbreak.info website has recently released data that it has detected B1617 lineage, that is the double variant's technical name, B1617, in India, Australia, Turkey, African countries, many European countries, UK, as well as in California. So it is spreading very fast. Now, if we look at the genome analysis and the mutations, if we map the mutations to the coronavirus genome, remember coronavirus genome is about 30,000 nucleotides. It is a single stranded RNA genome and it contains a lot of different proteins. The protein that is most important for determining the infectiousness as well as the virulence of the virus is the spike protein, which is shown here as S. These are the different variants that have been variants of concern recently popped up. So B117 is the UK variant, B1351 is the South Africa variant, P1 is the Brazilian variant, and B525 has been isolated from Nigeria, and now we have the Indian variant B1617. Now, as you can see that the virus actually has a lot of mutations, and even in ORF1A, these are different genes present in the coronavirus genome. Many of the genes, they have accumulated different mutations, and it remains to be seen. We need a lot of different studies to see how these affect the function of the coronavirus and whether it helps in improving the survival, infectiousness, and virulence of this virus. But we definitely know that spike protein is involved in getting entry into the host cells. So for B1617 variant, there have been two mutations that have been particularly of concern. One is L452R, we'll get back to the explanation of that in a minute, and the other one is E484Q. Now, as I think most of the students of biology are pretty aware, but in case normal regular folk are listening who don't have a background in genetics, mutation is any change in DNA or RNA sequence, any change in the genetic sequence. So these are the genetic changes that have been happening to the coronavirus, and these are the two specific changes that are present in the B1617 variant, the double mutant variant. We call it double mutant because of these two mutations that are important, but that does not mean that there are not other mutations. There are a lot of different mutations in this variant. So this is the list of characteristic mutations of this double variant. You can see that different genes have different variants. Even spike protein has lot of different mutations. So some of them may be neutral, some of them may be even harmful to the virus, some of them may be beneficial to the virus, may allow it to spread more fast or infect more people in a short span of time. So the two important mutations are E484Q and L452R. So in genetic terminology as well as biochemical terminology, this means that in spike protein at 484th amino acid, there is glutamate or glutamic acid. This is an amino acid, part of the protein, and glutamic acid is present in the normal wild type coronavirus. It has been replaced by glutamine. Now in terms of chemistry, just a little bit chemistry here, we have something which is COOH group, a highly acidic group, replaced by an amino group, an amine. Okay, so it changes the chemical reactivity as well as the chemical properties of that particular amino acid. The other one is L452R. So going back to the amino acid terminology, this means that this is amino acid number 452 in the spike protein, and we have leucine here, which is a very hydrophobic amino acid. And now it has been replaced by arginine. Arginine is a positively charged amino acid. Now, how does it actually affect the coronavirus infectivity? We need a lot of different studies to actually do that. We have not done those studies, so it is still uncertain how does that affect. But we know that these are non-conservative changes. These are changing the chemical nature of the spike protein. So what we know so far, the coronavirus double mutant strain has been named as B1617 strain. It is still only a variant under investigation. We are not sure whether it is of concern, which are the variants which are really dangerous, which have been proven, whose molecular mechanism has been studied in a little bit more detail. Those are known as variants of concern. So for example, UK variant, South African variant, Brazilian variant, they are variants of concern. But Indian double mutant variant is only a variant under investigation until now, not a variant of concern yet. So many mutations, as we have seen in the last slide, there are two specific mutations in spike protein, and that's why it is called a double mutant variant. The two mutations are E484Q and L452R. And this is the structure of the spike protein. So now we know from multiple structural biology studies that actually spike protein of the coronavirus interacts with this ACE2 receptor, which stands for angiotensin converting enzyme 2 receptor. So this is present in the lung cells as well as the lung epithelial cells, and that's where the virus actually gets entry into the host cells. So very, very important. So any changes which happen in this region, which either makes the binding strong, or in other regions which helps it to evade or escape the immune system, will help in propagation of virus, and they will be beneficial, and those virus variants will spread fast. That's how evolution works, and that is the cruel game of evolution that is going on. And this is a molecular model of how the spike protein, shown in orange here, interacts with the ACE2 receptor, shown in blue, and then it is engulfed by endocytosis, and then it enters into the host cell where it can multiply and cause its damage. Now this leaves us with the question what to do about this information. Of course, it is nice to know the molecular mechanism as well as the underlying mutations present in the coronavirus double mutant variant, but what do we do with this information? So as many experts in epidemiology as well as medical researchers have repeatedly said, the main course of action that most people, in fact everybody, should take are basically the same that have been enforced and suggested time and time again since the last year, and those are pretty much the same. Wear a mask, okay, maintain social distancing, wash your hands regularly, avoid going to any large gatherings, whether they are marriage functions, receptions, religious gatherings, any large gatherings, even public parks where you know there are a lot of people may be present. Avoid going to those gatherings and get your vaccine as soon as possible. See if you are eligible, and if you are eligible, then go to the nearest immunization center and get it done as soon as possible. Okay, in spite of the recent reports of the small risk of blood clots, the risk is so little as compared to the chances of getting the COVID-19 and later death, that it is much better, much less riskier to get the vaccine than not to get the vaccine. Okay, so this is about the coronavirus double mutant variant. I hope you like this information. If you have any questions, doubts or concerns, please let me know. If you like the video, please give it a thumbs up. For more informative video, please subscribe to my channel. Till the next video, take care and bye-bye.